Okay, I think we can start right now. If my audio is fine, I'm going to continue. So, welcome to all the participants. We have registered 152, and we hope to reach that number as we go along. So, this is coming from 38 countries. So, this is our second webinar about Gene Drive. This is this is entitled "Could Gene Drive Approaches Be Used to Protect Nature and Reverse Biodiversity Loss?" I am Dr. Dora Romero Aldemira, the director of the ISA Southeast Asia Center and the Global Knowledge Center on Crop Biotechnology. I am a trained scientist, a genetic engineer from Purdue University, and did my postdoctoral at Albert Ludwigs University in Germany. So, this webinar is hosted by the ISA Southeast Asia Center based in the Philippines in partnership with Outreach Network for Gene Drive Research. We also gratefully acknowledge the support of our ISA Biotech Information Centers, which you saw in the logos in the backdrop. So we have from Malaysia, China, Indonesia, Japan, Pakistan, Vietnam, Philippines, and Thailand. So the Gene Drive webinar series aimed to help promote a productive and balanced conversation on the benefits and risks of possible gene drive applications, providing factual and accurate information that can help place the discussion under the CBD context. Speakers today will explore the potential of gene drive applications for conservation, particularly to control invasive alien species which drive biodiversity loss and extinction of indigenous species. They will also explain the threat they pose, how researchers are seeking to address them through gene drive approaches, and the benefits and limitations of these approaches compared to more traditional control methods. This webinar will run for 1.5 hours, and we are currently on live streaming at the ISA FB and YouTube channels. All microphones are currently on mute, and questions should be entered in the Q&A function only. The chat function will be used by the panelists and for important messages to the participants. It's raining very hard here in the Philippines. Thank you all for responding to our pre-webinar surveys. And at the end of the webinar, we will provide a link to a post-webinar survey, and respondents will be given certificates. So let's me now introduce our moderator. Dr. Aroha Mead is an indigenous political scientist who works across the disciplines on indigenous rights and biocultural heritage issues. She is an independent researcher specializing in cultural and intellectual property rights of indigenous peoples, currently working with the Biological Heritage National Science Challenge and the Bioprotection Center of Research Excellence in New Zealand. I think I will stop there because it's raining very hard here. So let's move on, please. Dr. Arohami.
Kia ora. Thank you for the introduction, and greetings to everyone who's joined us this evening. Just to give a little bit of further background, I've been involved for probably 30 years in a range of initiatives where we've gone to a lot of effort to help explain new technologies, particularly involving genetics and more recently genomics, research to Indigenous peoples and local communities so that people can be informed in a safe space and able to make informed decisions about the implications of these technologies for them. I have the honor of being able to introduce to you all of the speakers today. And I'll start with Dr. Patty Bayo, who's the head of operations at Island Conservation based in Hawaii. She works to prevent extinction of native biodiversity, restore island ecosystems, and improve human livelihoods on islands through the removal of invasive species. Not everyone is familiar with the problem of invasive species, but in many regions of the world, and particularly in the Pacific. We have a lot of introduced species that are not indigenous to the lands, and they've created absolute havoc on the environments, and in many cases responsible for making extinct or putting in the highly threatened category some of our really prized indigenous species. So Patty will start, and then we're going to be followed by Dr. Owen Edwards, who leads a research group at Cicero in Australia, focusing on environmental genomics. He was also appointed leader of the environment and biocontrol domain of Cicero's Future Science platform in synthetic biology, where he oversees projects delivering environmental outcomes, including gene drives for biological control and engineering resilience in threatened ecosystems. And then our third speaker is from here from Aotearoa, Dr. Andrew Veal, who is a wildlife ecologist and geneticist at Manaki Whenua Land Care Research. And Dr. Veal's research interests span a range of fields such as invasion ecology, landscape genetics, evolutionary biology, genomics, animal behavior, and physiology. So I think you'll agree with me. We've got an incredible team of three presenting tonight. So let's start with Dr. Bio.
There. Hi, everyone. It's evening here in Hawaii, but good morning, good afternoon, good evening, depending on where you are. And let's see if I can start sharing my screen with you here. Did that work? Hold on.
It was working and then it stopped.
Yeah. Okay, let's try this one more time. Apologies. How is that? Are we back to working?
Yes, yeah. It's okay.
Okay, great. All right, so well, first of all, thank you very much for the invitation to be part of this great team. And I'm happy to share some of our experiences at Island Conservation with you guys, and to join in this very productive discussion around the potential role for gene drives in conservation. So I think what I will try to do today is to focus on invasive alien species and what are their impacts, what are the challenges that we face when dealing with invasive species, but also the opportunities that some of these, managing some of these species can represent for biodiversity and people alike. So I'm Patty, and I'm the head of operations for our US program based in Hawaii. And let's get to it. So I think the first thing I wanted to just go over a little bit is first establishing what are invasive alien species. For most of us, it's a concept we use frequently, but not for all of us. And invasive alien species are those that are not bad just because of their what they are, but for where they are. So essentially are species that have been introduced to outside of their native range, and in that new ecosystem, because of how that ecosystem works and the lack of control, natural control forces such as predators and diseases and other things, they establish and they overrule some of our native species. So in that context, invasive alien species can have huge impacts to native ecosystems. And as we know of today, they have been responsible for about 40% of all the extinctions that we know the causes for since the 17th century. So they have driven a lot of species to extinctions just by virtue of being in the wrong place. They unfortunately are still driving species towards extinction. So there's still a major current threat to most listed species that we have today. So they're adding a threat and a vulnerability to those species that are already at a brink of extinction. And as a new species in an ecosystem, they can have overall impacts that range from modifying structures to species composition, nutrient cycling, and all sorts of things at the ecosystem level. So they can have huge impacts for any ecosystem, any native ecosystem. As an example, here, about 80% of the threatened species in the Fimbos biome in South Africa are currently endangered by invasive alien species. So they can be a huge issue. And they cost human societies a whole lot. So this is a graph from a very recent publication that came out in Nature this year. And what they've done is to look at the largest data sets that there is, a database that has accumulated the most robust data set about the cost of biological invasions. And what you're looking here at these two graphs are just different ways to show the progression of these costs to human societies. So in the left, what you see is an annual cost in millions of dollars that starts in 1970 and goes all the way to 2017. And if you follow just the straight line here, you can see that it's a straight up progression of how much human societies are paying to manage the costs of invasive species. So this number here, the $83 billion, is the annual cost of invasive species in the 2000s. And this lower one here is likely an artifact of just how the data is collected, and it's unlikely that it has gone down. And rather, their estimate in the paper is that it has continued to go up, and it will continue to do so into the future. And to the right here, what you see in the two colors are two categories of costs. So one of them is just damage costs, and those are things like economic losses, yield loss, illness, infrastructure damage, and so on and so forth that have been linked to invasive species. And as you can see as well, the line just keeps going up as time goes by. And the management cost is how much human societies are spending to keep invasive species at a control rate. So keeping them from incurring some of the costs that you're seeing here. So the costs to human societies are very high when you look at invasive species, as well as to biodiversity. So there's both a biodiversity component, but also a very stark economic component to invasive species. And this is just another way to look at it, and just breaks it down by the types of invasive species. So you have here mosquitoes as one of the major, costlier invasive species that we've seen, rats, cats, and other things as well. So huge problem that we as a society have to manage. And as a society, we have managed this problem in many different ways. And I think it's often true that an ounce of prevention is worth a pound of cure. But in the case of invasive species, that is absolutely true. So the best and most cost-effective way to deal with invasive species is preventing their establishment to begin with. So preventing that new species arriving to native ecosystems is the best way forward. And we call that biosecurity. So it means that we are going to have a set of strategies that are in place to avoid the exchange of species between different ecosystems. And that is by far the most cost-effective way. When we fail to do that, the next best thing that we can do is to act in what we call the early detection and rapid response mode. So if a species crosses that biosecurity threshold and invades a new ecosystem, the fastest that you can identify its presence and the quicker that you can act to extirpate that small subset of individuals that have gotten there, again, the most cost-effective it will be. And so this EDRR is the next best thing short of preventing them from getting there in the first place. But that not always is the case. So in many instances, you're not even watching for certain species, and they get there and they establish themselves. And once they establish themselves, then you have two options. You have control efforts, which really means that you're just going to keep a population at a level that is reasonably low so that it minimizes the impacts it has in that native ecosystem. Or you can go towards eradications, meaning that you're going to remove every single individual of that invasive species from that native ecosystem. So those two are very different, and they have very different strategies associated with them. Control efforts, unfortunately, are very expensive, and they require a permanent and long-term commitment. So because you're just controlling, the minute you stop doing it, the species will continue to reproduce and reestablish. So you have to have a perpetual process that will maintain that population at low levels. And eradications are also costly, but when possible, a much better option, just because it's a one-time and you're done. You remove all of the individuals and then you establish biosecurity so that you don't have that problem in perpetuity. As an example of this, I think a good way to look at it is looking at islands as a model for what the issue is in the mainland. And islands are amazing. And as you all know, they are very special places, and island conservation focus on the removal of invasive species from islands to prevent extinctions. And the reason why we do that is that islands represent a very small percentage of the Earth's landmass. So their areas are very small, but they carry a very disproportionate amount of biodiversity and cultural diversity as well. So if you look at some of these numbers that are on the screen here, about 19% of all avian biodiversity are found on islands. And likewise, about 11% of all the world's population live and calls islands their home. So 25% of our world's nations are island nations. So they are a reservoir of both biodiversity, but also human and cultural values. But unfortunately, they've been the stage of extinctions. They're right stuck in the middle of the extinction crisis as we know it. So about 75% of all of the vertebrate extinctions that we know have happened on islands. And of those, 86% have happened because of alien species, invasive alien species. So if you take all of the extinctions that have happened on islands and you assign a cause to it, invasive alien species are related to 86% of those extinctions. And unfortunately, again, invasive alien species are still maintaining islands at that hotspot for extinctions, because about 41% of the highly threatened, the critically endangered and endangered species that we have are found on islands. And the presence of invasive species, again, is a major threat to those valued species. So what have we done about it as a community, as a conservation community worldwide? What you're looking at is a world map, obviously, and each one of those dots, and there's about 1,200 of them, represents an attempt to eradicate an invasive species from an island. And this encompasses all kinds of target species. So you have things like rodents, rats, and mice, and you have cats, and you have pigs and goats and other kinds of ungulates and whatnot. So this is kind of the overall eradications of invasive species on islands. And if you look at all of these species and these attempts to eradicate them from islands, we have a success rate of about 85%. So it's a very highly successful strategy that we've been implementing for many years. So it's well beyond the proof of concept. We know we can eradicate invasive species from islands at a very highly successful rate. And when we do that, the rewards are astounding. So when you remove an invasive species from an island, the recovery of the native ecosystem is quick and it's incredible. So that's kind of the the opportunity that islands represent, right? So this is the case, for example, of the Pinzon giant tortoise in the Galapagos Islands. So Pinzon Island had invasive rats, in fact, invasive rodents, including mice and rats. And the rats would predate on both eggs and the hatchlings of tortoises, of the Pinzon giant tortoise, preventing them from establishing and growing into adults. And so they hadn't reproduced in the wild for many years, for all we know, since Darwin wrote, the origin of the species. And but the Galapagos had kept a captive program of Pinzon giant tortoises for 50 so years, and they would hatch these little youngsters and they would raise them until they are rodent proof, so to speak. They're big enough that a rodent can no longer harm them, and then they would bring them back to the island. And an eradication project was done on Pinzon in 2012, and all the rats and all the mice were removed. And since then, we have seen natural establishment of hatchlings of the Pinzon giant tortoises for the first time on Pinzon Island for a very long time. So it's a very quick recovery that you can see in your time in your lifetime, which is often not the case with conservation interventions. They take a much longer ecological time frame to happen, and on islands this is just accelerated, and you can often see it much quicker. Another example is from Palmyra Atoll here on the Line Islands, and the Pisonia forest, which is a highly endangered type of plant. And again, rats were present on Palmyra, and they just predated on seedlings and prevented the establishment of seedlings of the Pisonia forest. And rats were removed in 2011. And what you see here is just a representation of the growth of the Pisonia forest as it in relation to my colleague here, Coral Wolf. And after only 6 years, once the rats were eradicated, what we had seen on Palmyra is a 5,000% increase on the Pisonia forest establishment on the atoll. So again, another great example of how rapid these outcomes, these conservation gains can be realized through eradication of invasive species. But the problem is that we have a whole lot more work to do. So what you're seeing now is a whole lot more dots. And what these dots represent are kind of the overlay of islands that have both threatened species and a current invasive species threat to those species. So those are islands that could benefit from eradications, that could benefit from this one strategy that we have found successful in many other places. The problem is that we can't really reach all of these islands with our current tools. So there is a huge need for innovations to help us address this issue and to realize some of those conservation gains that we've seen elsewhere. And as of it is now, if we look at just rodent eradications as an example, with the current tools that we have available, we can only reach about 0.3% of all of the rodent infested islands, because that's kind of where the limit of that tool is. And we can, I'd be happy to talk more about why that limit exists for this specific tool, but that is kind of an estimate of what we can do with the current tools. It's only feasible for us with those tools to address about 15% of the threatened species that could be helped by eradications on islands. So it's a small percentage of what needs to be done. And what really this means is that we're hitting that upper limit of the tool that we have available, because either the size of the islands, but more importantly, the complexity of those islands. And here I have just kind of the idea of how it's being done historically. And of course, innovation has happened, throughout the years when we've been doing eradications on islands. But we have followed this incremental innovation approach. And we need to break away from that incrementalism barrier. And the reason why is that the challenges that we have with eradications on islands are not just logistics and tactical challenges. As you increase the complexity of islands, if you increase the size of an island, the complexity doesn't just increase linearly. It's an exponential curve, because the moment you start getting bigger islands, you're going to have bigger ecological complexity and therefore ecological risk. You're going to have a much higher legal and regulatory requirements because of those risks. You're going to have the social political context with populated islands that have native communities, human communities that depend on resources and obviously will have a say on how these projects move forward. There's an ethical issue, there's a financial issue. So if you put it all together, the risks, they go up exponentially. The complexity of these projects go up much quicker than just a linear relation to the island size. So we really need innovations that can help us minimize risks while effectively still eradicating species from islands, invasive species. And this is just an example of these complexities from an ecological perspective here. So this is Floriana Island in the Galapagos, and we talked about how we eradicated bats on Pinzon Island, which was a smaller endeavor, still in the same archipelago in the Galapagos. But if you move to Floriana, it's a bigger island, so it's home to a much more complex ecological community. It also has a human community, a native human community to that island. So if you look at, our current tool for rodent eradications on islands is the use of pellets that contain rodenticide. So essentially you distribute those pellets everywhere on your island so that every single rat will have access to pellets and will receive a lethal dose of the rodenticide. But because of the complexity of the island, you will have the primary pathways of exposure as well as secondary pathways of exposure to a whole suite of other species that you don't want to harm, right? So your risks to non-target species here are just much, much higher. And for each one of these groups, you're going to have mitigation strategies in place to make sure that we're not harming non-target species in the process of eliminating the rodents. So the complexity is just incredible. So how do we do this? So what are the innovations that we're looking to help us address the issue of invasive species on islands and the impacts that they cause without increasing the risk to a level that is not acceptable by human societies? So one such way is to create tools that are species specific. So essentially a tool that would harm nothing else but the rat that you're trying to target. So if you can minimize risk to everything else in your ecosystem and you can target that species effectively, that would be the best of all worlds, right? You get rid of the problem, you don't harm anything else, your risks go down, your costs go down, your ethical dialogue is a much more straightforward dialogue. And one such way that has been evaluated or is being evaluated is the use of gene drives for eradications of invasive species on islands. And island conservation is part of a much broader partnership and program called GBIRD, which stands for Genetic Biocontrol of Invasive Rodents. And what it's trying to do is just to assess the feasibility of having a genetically modified mice that can be introduced to an island and you're all going to hear a lot more about gene drives and how they work and what are the issues related to that. But could we have a mice that is genetically modified in a way that would drive a rodent population on an island to extinction without affecting non-target species to the degree that we know our current tools do? So I'll just leave you with that. And I think that will be a good segue to our following speaker, Owen. And thank you all so much for the opportunity to speak today about the work that we do and the experiences that we've had. And I'm happy to answer any questions on the work that we do. So mahalo nui noa, as they say in Hawaiian, thank you very much. And I look forward to hearing from the other panelists.
Oh, thank you very much. I'd just like to remind all participants that if you want to ask questions, please do so in the chat, in the Q&A box at the bottom of the screen, and our speakers will gladly answer any of your questions during the session. Thank you, Patty, for that clear explanation of the scale and scope of threats posed by invasive alien species to conservation and biodiversity, and particularly like that you were able to show some positive examples of where invasive species have been eradicated and ecosystems restored. Now I'd like to invite Dr. Edwards to tell us a little bit more about how these threats can be addressed using gene drives and why these approaches are being explored. So, Dr. Edwards, you have the floor.
Thank you very much. Are my slides visible? Yes.
Yes.
So thanks for the invitation to speak today as part of this panel. I'm going to be giving a bit of an Australian perspective in terms of invasive species management. I work for CSIRO, as was said before, which is the National Research Organization. And this is actually a partnership with a couple of local universities, University of Adelaide and Paul Thomas, who spoke at the last webinar series event, and also Maciek Maselka, who's at Macquarie University in a joint appointment with CSIRO. Oops. So invasive species in Australia, I'm particularly interested in we have significant problems with invasive animal species in the country. They impact on our endemic fauna and also obviously cause biodiversity loss. The most impactful of these are shown here, some of the more famous ones in terms of the cane toad, but the fox and the feral cat in particular as predators are the ones that are having the greatest impact. And following on from what Patty was saying, I'll just show a slide here that shows on the top graph is the threats, the factors that have caused extinctions of mammals in the past. And you can see that feral cats are at the top of the list. Foxes and rabbits are the next two most important factors causing previous mammal extinctions. But at the bottom, for the current threats to extant mammal species, cats remain at the top of the list, as do foxes in third place. And just to give you an idea of the impact of feral cats within Australia, these are the numbers of birds killed by feral cats in Australia each year at the top, and the number of reptiles killed by feral cats in Australia each year. So the numbers are truly mind-boggling. We're also dealing with a mouse plague in Australia at the moment. And in this case, the mice themselves are not necessarily causing biodiversity loss, but as Patty was saying, we get indirect effects as well, because the only way we can manage these mice is with chemical baits. These are released to kill up to 25,000 mice per hectare at the moment. And so you can imagine across hundreds of thousands of hectares how much chemical is going out there. And we know already we're seeing impacts on predatory birds like the powerful owl. And we have expectations that we will see impacts on mammalian and reptilian predators as well, like the eastern quoll and the goanna. Thankfully, just yesterday, our regulators rejected a bid to use bromadiolone, which is a anticoagulant rodenticide against mouse plagues because of the fears of the threats for our native wildlife. Now, Australia has attempted to use biocontrols such as viruses for invasive animal control in the past, and there have been well-known successes, in particular with rabbits. This is a graph that's shown here with 3 different controls: myxomatosis, rabbit fleas, and calicivirus or rabbit hemorrhagic disease. But these successes are not permanent. Biological control is often preached as something that has the potential to be permanent. In this case, what we see is an immediate effect that drops the rabbit populations below the damage threshold, which is the red line near the bottom of the slide. But we do see resistance occurs within about a period of usually about 8 to 10 years, and the populations rebound. And so it's necessary to find a new biological control to release. So this is not, in history at least, a permanent solution for invasive animals. And my organization, CSIRO, has also been at the forefront of developing genetic control strategies against invasive species since the 1950s and 1960s. Many of these involve the release of sterile or otherwise incompatible males, which compete with naturally occurring males for females and result in sterile offspring or sterile eggs being produced, eggs that don't develop, and therefore you see population suppression. And my colleague Maciek Maselko has recently developed more sophisticated approaches using this sterile male. This is based on CRISPR-Cas9 transcriptional activator technology. So it's a modified genome editing technology. And this works for multiple generations. So it's better than previous types of sterile male control strategies, but you still have to rear and release extremely large numbers in order to have an impact on your target population. And that might be possible for insects like mosquitoes or fruit flies, where you can rear large numbers in factories, but it's just not conceivable that you can use this sort of a genetic control, that you can rear sufficient numbers to control massive populations in the field. Well, gene drive is a genetic control technology that has the potential to overcome this issue of having to rear massive numbers. Without a gene drive, as is shown on this slide, if you release an individual carrying a genetic control as a transgene into your target population, generally the trait will become diluted and the frequency of that trait will diminish each generation. Whereas if you release a gene drive driven genetic control into a population, instead you see an increase in frequency each generation until the entire population can be affected. And this is done by biasing its inheritance. This is traditionally done, or most commonly done these days, with what's called a homing gene drive mechanism. When inherited from one parent, the gene drive copies itself onto the matching chromosome inherited from the other parent, which means all of the offspring from the cross will inherit the genetic control. The homing construct includes Cas9 and a guide RNA for precision cutting of the chromosome, and a cargo, which usually represents the trait that one wants to drive through the population for population suppression or for population replacement. Homing drives have been demonstrated to function very effectively in some insect systems, like mosquitoes, but so far nobody has been able to get this sort of a drive to function effectively in vertebrates. As a result, we're working with Paul Thomas, who spoke at the last seminar series event on technologies such as this one, the X chromosome shredder, which does not rely on this homing mechanism. And the way this works is that the Cas9 and the guide RNA target repeated sequences on the X chromosome. And so when this is expressed within the testes, it basically eliminates any of the sperm that are carrying the X chromosome, and in doing so biases the population towards males. Paul in the last event showed modeling which showed how effective this sort of an approach can actually be. But a highly effective gene drive has the potential to drive a particular control through an entire population from a single release. This potential for spread also increases the risk associated, or the risk of unintended ecological consequences, such as spreading to areas where an invasive species is actually endemic, or even regulatory issues such as spreading across borders into other jurisdictions. For this reason, we are interested in molecular switches that turn off the gene drive, making it self-limiting in space and/or time. Our collaborators at the University of Minnesota in the US have been testing a generation counter technology that we hope will allow us to tune a gene drive to switch off after it has achieved its desired impact. And not going into the details of the molecular mechanism, basically the technology counts every time that it is passed from one generation to the next. And so once the population has been fully affected, you can tune the gene drive to stop at that particular time. And that prevents the impact on a target population, but can prevent the impacts on non-target populations. As I mentioned, in terms of applications, there are two types of gene drives that are being considered. The first, and what most of the conversations that we've had to date are about, are called suppression drives. And these aim to suppress populations by driving a trait that impacts on reproductive fitness in some way. Our approach for suppressive drives aims to manipulate sex ratio or to create a strong male bias, which causes populations to eventually collapse. An example of this has been demonstrated in the laboratory for mosquitoes, which targets a double sex gene, which controls sex determination. And by driving this particular trait through, it again biases populations towards males and causes the populations to be suppressed. This is a sort of drive that we're interested in for things like mouse plagues, for carp in our waterways, European carp, and for agricultural pests, such as spotted winged Drosophila, which isn't in Australia yet, but is a strong threat of it being introduced. The second type of gene drive is referred to as a replacement drive and aims to drive a trait through the population to make it less harmful. This has also been demonstrated in the laboratory for mosquitoes, in this case, driving a trait that prevents the mosquito from being able to transmit malaria. We are interested in this sort of approach for a species like the cane toad, which produce a toxin and affect the predators by killing them, any that try to feed on the cane toad. And so we're interested in using a drive that knocks out a gene that's associated with the production of the toxin, so that we can potentially replace toxic cane toad populations with non-toxic cane toads. So there are some advantages of the gene drives which make it an attractive technology. As I said, it can overcome the costs and logistical issues that limit the use of current options, such as chemicals, as Patty was referring to, and this includes genetic controls, as it's not necessary to rear up the large numbers, it's self-spreading. The modeling shows that the gene drives can represent a permanent solution for some target invasive alien species. As Patty also referred to, if properly designed, there should be no direct effects on non-target species. The sequence that you target can be specific to the target species, so that even if you have hybridization, that gene drive will not spread in that other species. And finally, they can be designed such as manipulating sex ratio to be much more humane than existing management options. And it's for this reason that when we recently got funded to try and develop gene drives for mouse plague control, PETA Australia, the animal rights group, actually came out and said that this is a much more humane approach in their mind. Even though it's using genetic modification, it's a much more humane approach for controlling mice compared to the current chemical strategies. Now, there are limitations to the use of gene drives. We'll be talking about regulatory and public acceptance type limitations in the next webinar series. But in terms of just the technology itself, the speed of control depends on the generation time. And so for those animals that have a very long generation time, you cannot expect for control to happen for several decades. And so that may not be an acceptable control under some circumstances. It's also true that the released individuals must be reproductively competitive after you release them. And so this can be a challenge when you have to rear your individuals in the lab to generate the gene drive, and they become adapted to laboratory conditions. This can impact on their effectiveness after you do release them. So you have to develop strategies to allow those individuals to remain reproductively competitive. And I'll just mention that we have developed a tool that allows us to assess some of these limitations as well as the advantages just by looking at population genomic data from target species. It allows us to basically analyze and ask the questions about whether it's feasible to do releases, how many individuals would need to be released to control over what period of time, based on the populations that you select, but also based on the targets that you might choose for your gene drive. I think it's important for us to recognize the status of gene drive research at the moment. Functional gene drives have only ever been tested in insects in laboratories or in specialized containment facilities, first in the model species Drosophila, and then later, most recently, against mosquitoes. There have not been any that have been approved for release into the environment as of yet, even just for testing purposes. And in vertebrates, no functional gene drives have as of yet been developed. We're still working on them. And all current gene drive research in vertebrates is being conducted in model laboratory animals, either in mice or in zebrafish, at least to my knowledge. So it's still a ways off. There's still a lot of research to be done. And there's a lot of discussion, like the forums such as this one, that should be had before we even consider doing any releases. So my takeaway messages then from this talk: I think gene drives should be considered as part of a portfolio of management approaches for invasive alien species, together with existing chemical, biological, and genetic control options. I say this because gene drives are not and are never likely to be a silver bullet, but for some targets, they will be perhaps an ideal management option. For others, we will still have to rely on some of the conventional approaches. Gene drives do provide an option to spread genetic control technologies that already exist that would otherwise be infeasible because of this issue of scale. It allows also us to target populations that are inaccessible or very difficult to access through roads or planes, etc., which is another advantage. And finally, gene drives, it's possible that they may increase the risk of unintended consequences, but these risks can be mitigated using technologies that make them self-limiting in space and time. And so I think it's equally important that while we work on developing effective gene drives, we also focus on developing technologies that will help us to tune these gene drives so that they only achieve the outcomes that we want once they're released. So I'll just finish by acknowledging again Paul Thomas, who spoke previously in this series from the University of Adelaide, and Maciek Maselko, a joint appointment between CSIRO and Macquarie University, who are the true engineers of our gene drive options, our genetic control options, as part of the broader consortium that we're a part of within Australia, and also with the GBIRD consortium that Patty mentioned at the end of her talk. So I'll be happy to answer any questions that you might have in the last part of this webinar.
Oh, great. Thank you very much. I'm not going to be putting my video on because my internet connection is not very good, but it's Aroha here, the moderator. So I really want to thank you for giving us that overview of gene drive approaches to preventing biodiversity loss that's currently being explored. I'm sure that there'll be a lot of questions raised by your presentation, because it's quite a mind-boggling type of technology to get one's head around. I'd now like to invite our final speaker, Dr. Andrew Veal, to give us an idea of the scope and also the limitations of these approaches. So, Dr. Veal, could I invite you to present, please?
Hi, everyone. Hopefully
Owen,
you can hear me.
Yes,
can you unshare, please? Unshare your slide.
I'm trying to, I'm sorry. I've lost the,
There, okay.
thank you.
Has done it. So can you see my slides?
Yes, we can, thank you.
Excellent. So, kia ora everyone. I am a scientist at Landcare Research in Aotearoa, New Zealand, and I'm here to talk about the potential usefulness and limitations of gene drives approaches in mammalian control in New Zealand. Now, in New Zealand, we have this movement called Predator Free 2050, which is now also a company funded through the government, and we are hoping to remove rats, stoats, and possums from the entire country. Now, the size of New Zealand is about the same size as the UK, and it's often discussed the potential for using gene drives in this quest. So this is effectively removing all these pest species from a very large country in 30 years. Now, New Zealand conservation ecology is different to continental systems. We have very high endemism, and we have island gigantism and flightlessness of our birds. We only had 3 native mammals prior to humans arriving in 1280 AD, and they were all bats, and one of those has since gone extinct. So effectively, it was an island of invertebrates, birds, and reptiles. Since humans have arrived, we've had 31 species of feral exotic mammals arrive. Over 40% of the birds in this 800 years have gone extinct, and a third of the birds currently in the country are threatened. Introduced mammals are the primary agent of this decline. And you can see here, so Polynesians arrived around 1280, and they brought the Polynesian rat, Kiori, and also the dog, Kuri. And they also hunted a lot of the flightless birds to extinction. And then Europeans arrived from effectively the 1600s. And rapidly we had lots of different predators arrive, and then lots of birds went extinct. And these are just some of the birds that went extinct in the last 100 years. So effectively, New Zealand is currently in the middle of a mass extinction event. And this is entirely caused by introduced mammals. What this means is that conservation in New Zealand means killing small furry animals. And I would say that basically the only meaningful conservation for most terrestrial species is killing pest animals. And we do this through trapping. We do it through a lot of poisoning. And a lot of that poison is effectively through monsoon buckets. So we coat the entire area in poisons, either acute toxins or anticoagulants. And obviously there's quite a lot of social opposition to this, because no one wants to see the environment covered in poison. However, this is the only way that many of our native birds can actually survive. Now, the target species for Predator-Free 2050 are, as I said, rats, stoats, and possums. Mice are not actually one of the official targeted species, but they are also a big pest and is something that we need to discuss. So we've got these 2 European rats, the ship rat, Rattus rattus, and Norway rats, Rattus norvegicus. They are omnivores, they are nest predators, they predate on, they eat a lot of seeds and change the canopy through that, and they also affect a lot of invertebrates. The stoat, that was a carnivore that was introduced to kill rabbits, and they didn't know what their job description was, and so they went into the forest and killed all the birds. And the possums were introduced to start a fur trade, and they also then immediately rapidly expanded, and they're also nest predators, as well as they selectively browse specific tree species and can cause canopy collapse. So these are what we're hoping to get rid of in the next 30 years. And if we don't control these species, indigenous bird species will go extinct. Now, because we need to control these at huge landscape levels, gene drives have been proposed as a potential solution. Right now, of course, we're relying on toxins. And for all of these species, no large-scale eradication program has ever succeeded without toxin. We need a lot of toxin, and toxins are not species-specific. They have off-target effects, and there's social opposition to their use in this way. And there's really no true tool that we have to control most of these species at these scales. And so potentially, obviously, gene drives are very species specific and could be a solution to the problem that we face. Now, there are limitations for gene drives, specifically in New Zealand. Obviously, there's a question of social acceptability of gene drives and who gets to decide. You know, if 51% of people say yes, is that enough? And that's a very complex question involving Maori, involving people in New Zealand and around the world, because for some of these species, particularly the rats, they could spread outside of New Zealand and then cause problems elsewhere. There are technical challenges in creating the gene drives. As Owen said in the previous talk, we don't actually have a working gene drive yet for mammals. And we've currently got a 30-year time frame to make one, perfect one, test one, and spread it through a huge country. Also, there can be resistance develops. Species could interact, so you might be able to remove one species and then have mesopredator release. And there could also be extinction recolonization dynamics, where basically it flows through populations in a spatial way, but then they reflow from other areas. The big issues that I see are life history problems in terms of generation time and breeding facility logistics. So all of these things need to be discussed. Now, life history is the one that I really wanted to point out. These are the minimum generation times for the species that we're talking about. While weasels and ferrets are pests in the country, they're also being discussed as potential ones to get rid of. Now, for almost all these species, we have excellent genomes. This is something that Predator-Free have been investing heavily into. And so we now have a novel stoat genome that I helped work on. Possum genome's recently been created. And the shiprat genome has also been created. The Norway rat and the mouse already exist. So that's a good thing. We have the genomic information. Unfortunately, the minimum generation time for particularly stoats, ferrets, and possums is very long. So for males of these species, it's a minimum generation time of 2 years. That's very different to the 8 weeks for mice and rats. The life expectancy of them is also high, particularly for possums. They can live up to 13 years. Now, this causes a big problem in terms of the rate that these things can spread. This was some modeling that has been done in collaboration with Thomas Prouse in Australia. And if we start with a population size of 10,000 at the start, we can cause rapid population extinction of both mice and rats with reasonable breeding facilities producing the gene drive rodents. For stoats, and this is optimistic because this is based on a 1 year generation time rather than a 2 year, which is how male stoats work. We don't have population crash for 30 years at least. This is if we created a perfect gene drive and released it tomorrow, we don't make 2050. With possums, given that they can live up to 13 years and they've got a minimum generation time of 2 years, we're talking really 100 years minimum in a perfect scenario where everything works. So overall, gene drive for mice now, this is being worked on in the US and Australia with the X-shredder. And it's got a sequenced genome, it's economically important. Potentially an X-shredder may be working in the near future. It's being worked on, as I said. And there are still questions. So can these modified mice invade a population? How long will it take? The social acceptability is a big one, and high risk of spread beyond New Zealand. But I'm quite hopeful that a gene drive in mice will be created. And so this is really the proof of concept. If we can get mice working, and as I said, it's probably happening not in New Zealand, but in other countries, this could be a really exciting development for biodiversity conservation in New Zealand. Now, rats, they're kind of like big mice. And so there's one method that might be possible in mice that's a T element. They don't have it. But any of the other ones, such as the X-shredder or homing endonucleases, if we can get it working in a mouse, it's very similar for the 2 rat species. again, there is a risk of spread beyond New Zealand. And so we do need these mitigation measures so that we can target just them spreading in New Zealand, or just targeting genetic variation that is within New Zealand and not elsewhere. But again, this is a hopeful situation. They've got a short generation time, and if we can get mice working, don't see any reason why we couldn't for rats. Now, stoats, they're very tricky. So female stoats are actually impregnated shortly after they're born, and then the eggs are just sitting there fertilized for a year doing nothing in diapause. However, the males aren't fertile until they're a year old, which means that for males, it's a 2-year minimum generation time. So far, we basically can't breed them in captivity. They are highly stressed animals, and so we just can't actually breed them in captivity. And we'd really need to create thousands of these per year in a breeding facility, and we don't actually know how to breed them in captivity. So realistically, it's a minimum, I would say, 70-year time scale that if we worked really hard on this. Currently, there is no work on creating a gene drive being done for stoats. Realistically, I think that we're more talking about a 100-year time frame. Now, that's not probably good enough for most species, so we do need to be working on other things, or just creating, but continuing control, so that we can save the species until we have a tool that will work. The possum, minimum 2-year generation time, and it's generally 1 offspring per year. And my understanding is that we can't currently CRISPR the germlines of marsupials. And we would need to create thousands per year, ideally. And each of these takes up probably a few square meters of a breeding facility. So once you get 3 square meters times thousands of individuals, then you're starting to look at huge breeding facilities. And the logistics of that are quite challenging. Again, minimum, if everything worked perfectly and we had magical technology to just do this, it's a minimum 70-year, and really it's likely to be over 100 years. So in terms of what will happen, it's likely that if we depended on a gene drive for possums, they'll be here longer into the future than they have been in the past. Now, there are lots of technical challenges even for mice. And so this is about the individual fitness, whether they can be connected across that landscape, and how well it will spread. So my predictions for gene drives being useful for conservation in New Zealand is, I really hope that mice which are being worked on in other countries, hopefully we have a laboratory proof of concept of a working gene drive within 5 years. And it would be great that if we had field trials within 10 years. And once we get mice, as I said, rats, it could happen. So this is really promising. And I really hope that we do continue having the conversation about the ethics and the social issues about this, so that we can look at this compared to other methods. There is no way that we can get rid of these species without magical future technology, and this is a potential magical future technology that might work. Now, really with stoats and possums, we are talking about very, very long-term things. Now, with possums, maybe because they breed slowly enough, we could get rid of them with current technology invested in with probably billions of dollars. There is no way that I can see getting stoats out of the country for the next 100 years. So even though we've got this delay, probable or definite, for a gene drive method for eradicating stoats from the country, I can't actually see another way that we can eradicate them. So I don't want this to seem a negative thing. There's very few things that could potentially do this. I just wanted to note, there is no work currently being done creating gene drives in New Zealand. A lot of people are very concerned about gene drives, but we are doing the fundamental work of sequencing the genomes and modeling how these might work, so that we can discuss these things and see if it can help conservation and what the timelines actually are. So I think that's me. I'm very happy to have any of your questions. Thank you.
Oh, kia ora, Andrew. Thanks very much for that. And in particular, for being able to provide us just with some really pragmatic and comparative assessments of what these approaches could likely achieve, while noting that you have a great deal of optimism about the potential of the tools to meet conservation objectives. You also noted that nothing is currently at field trial level yet. So now we have time for some questions and answers, and I can see that many of our participants have been busy typing away in the question and answer box, and some 6 questions have already been answered, but this is an opportunity to just be able to address some of the other questions as a whole group as part of the session. So somebody has asked in both the chat and in the question and answer something that they obviously want to know about, and that question is, globally, which kingdom of species has cost the environment more than the other? Is it the Plantae kingdom or Animalia kingdom? Would any of our speakers like to respond to that question?
I think I attempted to answer, but I might have done it privately. And I think that it depends on what you're looking at. If you're looking at island communities, for instance, for certain the vertebrate invasive species, predators in particular, are what's going to cause the most ecological damage. So in generally speaking, invasive predators are going to be more, they're going to have higher ecological impacts. That being said, things like invertebrates, such as mosquitoes or agricultural pests and whatnot, have huge impacts, as was demonstrated in some of the graphs I had on the talk. So it really depends on where you are, if it's continental or an island ecosystem, and what kind of damage you're focusing on, if it is economic losses or biodiversity losses. You can have a very different approach. And of course, I mean, they're all bad, essentially. Plants can also have a very high impact on many ecosystems, and they modify nutrient cycles and whatnot, and can create havoc as well on native ecosystems. So I guess my answer is they're all bad, depending on your context. Some will be a little worse than others.
Thanks for that response. And now Farshid Talat has asked, what if undesirable genes escape from target plant or animal and make serious problems to normal ecosystems? Would someone like to respond to that?
So it's Owen Edwards here. As I sort of alluded to in my talk, I mean, it both in terms of any sort of horizontal transfer or hybridization, it's absolutely the case that if the gene drives are designed properly and designed to target sequences that are very unique to the target species, then even if that gene drive escapes through horizontal transfer or through hybridization into another species, it won't have the function of being a gene drive. It won't have a sequence to target. So it will not spread in the same way in another species as it would in the target species. But of course, that does depend on appropriate design of the gene drives, and that's why it's so important that we aim to do releases into countries that have very strong regulatory regimes that will look very stringently at the design of these gene drives to make sure that the questions such as the specificity of the target are addressed, and the evidence is produced that that sequence is not present and is not likely to present in any other species within the environment.
Thank you very much for that response. If hopefully that answers your question, and if it doesn't, just I'm sure it does, but if there's any follow-up, please indicate in the chat. So a number of people have asked a question around how you as a researcher and all your organizations are approaching engagement with the public, and specifically with Indigenous peoples. I'd like each of you to answer that, although Patty, I know you already answered in the chat, but if you could, just for everyone's benefit, just explain to us how you are dealing with public engagement, and specifically with Indigenous peoples. Thank you. Maybe we'll start with you, Andrew.
Okay. So Manaki Whenua hasn't been involved specifically in any gene drive research, given that currently there isn't an appetite politically to look at that. Therefore, I haven't been specifically involved with engagement other than some meetings or hui with various iwi that were arranged through Kevin Esvelt's group, who came to New Zealand. So this is one of the big international names in gene drive research. And so I've been present at those meetings, and there is quite a lot of engagement in New Zealand with that, and there needs to continue to be. But yeah, I haven't been highly involved in that, given that we're not actually doing gene drive research specifically yet.
Right. Owen, would you like to give a response?
Sure. So as it was at the beginning of my slide, the gene drive research is being conducted in CSIRO, at least with our collaborators, is being sponsored by this platform called the Synthetic Biology Future Science Platform. I lead the environment and biocontrol domain within that. We have a domain that's entirely focused on addressing the issues of public acceptance for gene drives and other synthetic biology technologies. And so in parallel to the research that we're doing in terms of addressing the ecological and technological questions, we have research that is ongoing, including surveys of the Australian population that have already been conducted, asking questions about the acceptance of gene drives, as well as other synthetic biology technologies that are being proposed to make ecosystems more resilient. So yes, we have an investment. We haven't addressed Indigenous issues to date specifically. Part of the issue within Australia is that Indigenous communities are so diverse across the country. Until we actually know where we are going to target, it's very hard to generally address the Indigenous issues. But we have just appointed somebody within this part of our Synthetic Biology Future Science platform who is focused entirely on Indigenous issues. So it's something that we are about to address.
Great. Okay, Patty, would you like to just share with all the participants how your organisation has been progressing your work with public engagement and with Indigenous peoples in the islands?
Yeah, absolutely. So I think for island conservation, meaningful social engagement is paramount in all of the project phases. In the context of the US, where I do most of my work is focused on, we have a very robust regulatory process that mandates public engagement. So there are venues and forums that are legally established for those engagements, but we all know that those are not necessarily the best forums, and they're not necessarily sufficient to provide true engagement and meaningful engagement. So I see strives to engage communities as much as we possibly can. And our overall strategy is one of transparency and sharing accurate information so that both the potential risks and the potential benefits are understood, and folks can make informed decisions about whether or not it is the appropriate intervention that serves their set of values and not just ours. So I think we come in with a set of values that is shaped by our biodiversity protection values and whatnot, but that has to be matched with local values that see the project as an important piece as well, in achieving the outcomes is important for that community as well. So we do strive to do that in every project, and we succeed and we fail in that endeavor, but we do strive to do that. And as it relates to gene drives in particular, the program that I mentioned earlier on the potential genetically modified mice, one of the main components of that program is the social and ethical piece of it. I think one big question is obviously, can we do it from a technological perspective? Can we produce a gene drive? Can we make it work? Can we restrict it? And so on and so forth. But I think a second question that is addressed in that group, and we have specialists looking at it, is should we do it? Is it something that we should, as a society and in specific places, should we go ahead and do it? So I think that kind of guides our approach to all projects. And I feel it's one of the most, if not the most important thing that we do early on in projects as we're setting out.
Thanks very much for that very considered response, Patty. I know that here in New Zealand, just building on Andrew's response, the fact that the technology and this type of research can't be applied in the field yet actually provides a really good space to have a conversation because there's no urgency, as in people aren't being asked at a meeting to give acceptance or not, which has happened in the past with previous types of new technologies. So we've got a little bit of time to have considered discussions and have people really consider the issues and not feel pressured to have to make a decision at this particular point in time. So just coming back to some of the questions of the participants, another one here is, can gene drive approaches be also used in eradicating or controlling pathogenic microorganisms, bacteria and viruses, especially those that are multi-drug resistant? Now, who would like to respond to this question?
So I can say that so gene drives will only function against sexually reproducing organisms, and so most pathogens, at least bacterial and viral pathogens, cannot be directly targeted. But there are approaches that are being developed that aim to control pathogens by driving a resistance trait through the host, or I mean, similar to mosquitoes, they're trying to control malaria by making the mosquitoes not capable of transmitting it. Strategies like that are the only way that I can think of that a gene drive might be used to target a particular pathogen. There are naturally occurring gene drives in fungi, which one of our researchers at CSIRO are investigating and considering that they might be used for control of fungi in the future, but not bacteria or viruses, I don't think.
Thanks very much. And another question is, what is the progress of gene drive technology for controlling alien invasive plant species? And one is named, so I don't know it, so excuse my pronunciation, Parthenium hysterophorus, which has devastated biodiversity on many continents, including Africa and Asia.
So it's Owen Edwards here again. There are a lot of people that are very interested in the potential for gene drives, including researchers for control of invasive plants, including researchers in my organization. The first issue or technological issue is the issue that exists that has prevented the development of the homing-based gene drives in mammals. That issue is even worse in plants. And so that's a challenge that they would have that has to be overcome in the first instance. It doesn't appear like the X-shredder option has an analogous one that could be used in plants, unfortunately. There are also aspects of plant biology that make plants a difficult target. The only ones that would really work effectively, I think, are obligate outcrossing species. And so I don't know whether the ones you've mentioned are in that category, but obviously inbreeding ones would be very, very difficult to target. The other issue is seed banks. The seed banks provide basically a defense against gene drives in plants. So a gene drive could pass through a population, and then in the next generation, the seed banks would just grow, and you'd have the plant would be back again. And polyploidy is another issue that exists and is quite widespread in plants that has not been tested yet in terms of how effectively a gene drive could work in polyploid species. So I think it will happen, testing of gene drives in plant species, but for the moment, I think it's likely that they'll be looking for examples of effectiveness in more tractable systems first before we'll see a lot of activity in plants.
Okay, thank you. Well, I've been given the word that was the last question. And so what I'd like to do now is just recap some of the takeaway messages from each of your presentations. So with Patty's first presentation, some of the key messages that she shared with us were that invasive alien species are a serious ecological, social, and economic issue, and that management of invasive alien species is costly and has been largely inefficient to date, that islands represent an opportunity for the eradication of invasive alien species, and that incremental innovation will not help meet the challenge. It requires something more urgent than that. Dr. Owen Edwards, in his presentation, sort of reminded us that gene drives need to be considered as part of a portfolio of management approaches for invasive alien species, together with existing chemical, biological, and genetic control options, and that gene drives provide an option to spread genetic control technologies that would otherwise be infeasible because of scale. And he also indicated that gene drives increase the risk of unintended consequences which can be mitigated using technologies that make them self-limiting in space and or time. And Andrew's presentation sort of raised that there are a lot of technical challenges to eradicating pests beyond just the genetics, but expressed optimism of the ability of gene-drive tools to assist in conservation goals. So I think it's been a really rich session, and I'd now like to hand over to our final speaker just to conclude our session, to Maha Arujanan, who's the Global Coordinator of ISA, and leave the final words with you, Maha. Thank you.
Thank you, Aruha. It was really enriching and a very informative webinar. You know, what is really amazing here is where scientists come together to discuss both the advantages and limitations. And this is where we believe trust and transparency is developed, two values, strong values that ISA stand for. ISA is very happy to be part of this initiative where a platform is created to discuss this very important topic and tool. And we all know that gene drive is a very new technology, and we need more understanding on the science, its potential, risks, and how these risks are managed. More importantly, making sure that the right messages are conveyed to all our stakeholders. What is really interesting is how the technology actually go hand in hand with biodiversity and conservation to address the negative impact of invasive alien species. I'm not going to go into the summary again. Aroha did a very good job, but I want to say that as a science communicator, it is so important for us to frame our messages in the right way from the beginning and use the right examples. And webinar like this help to consolidate those messages from scientists, and then everyone speaks the same language. This, I believe, helps in the adoption of the technology and science-based regulations. And that is the aim of the webinar series. So the previous webinar, we finished one so far, could be watched on ISA YouTube. This webinar, current one, will also be posted there. So please share the link with your colleagues and network. I don't want to take so much of your time. I want to thank everyone, all the speakers, audience, our partners, and also Outreach Network for Gene Drive Research and ISA Biotechnology Information Centers, and of course, my team at ISA. Thank you so much for your presence, for all the efforts. Tune in for our webinar number 3 on July 8th with yet another very important topic, that is how gene drive controls vector-borne diseases. So till then, stay safe and stay connected. Thank you very much.
Thank you so much, Maha. We're still not over. We'd like to ask our participants to look at the chat box. We have the post-webinar survey link posted in there. So we'd like to get all your comments or your evaluation or any questions that come up that we haven't answered, and we'd like to hear from you. So again, thank you so much for being here with us. Thank you, our very, very prestigious panel of speakers. I learned a lot. Actually, this is the first time that I have heard about invasive species, and I'd really like to ask questions. I may email you because I don't have time to ask questions. And yeah, I think we're ready with our last, the next webinar. We have a backdrop on that yet. EJ, can you post that? Can you post our next webinar? We already have made the poster for the next webinar, webinar 3. So we will be sending this out in our social media, in our email. So this one. So it's going to be, can gene drive eliminate vector-borne diseases? And the registration is now on as soon as this is out. And so we'd like to receive more registrants. We know that we have gone up again in our registration and our participants during this webinar. So thank you once again, and have a good day. Take care, be healthy, be safe. Bye-bye.
Mahalo.
For those who are still here, you can look at the YouTube channel of ISA. ISA YouTube channel. The Zoom video is going to be posted there as soon as this is over. Thanks.
Thank you.
Thank you, Andrew.
See you.
I was muted. Thank you, Andrew. Thank you so much.
Thank you, everyone.
Oh, wala na sila.
Ola, let's just stay back. ISA team.
Okay, I gotta go.
You have to go?
No, I gotta go. 1 Minuten, bitte.
Okay, I just want the ISA team to celebrate EJ's birthday.
Ma, every time I see your bookshelves, I'm trying to read the titles of your books.
Yeah, it's not really arranged. The other day, do you know, Clem, the book called Don't Be Such a Scientist? The other day I was looking for the book. Finally, I found it. Yeah, a lot of it
Wow.
is, it's all mixed. It's management, insurance, Chinese books. Yeah, and I've got some books, TED Talks, and it's all mixed.
Yeah, but you have a very large selection there.
Yeah. I see Violeta. How do you pronounce it? Violeta there. What's the name? I forgot her big nickname.
Okay, everybody out yet?
It's
Okay.
just
Yes.
And I celebrate EJ's birthday too.
Oh,
Yes.
happy birthday!
That's EJ. Put EJ on the spotlight. I see. I don't know whether it's only me or I see myself on the spotlight. Can we put EJ
You
on this?
are.
Yeah. Okay. Go, oh yes. Okay. Let's sing EJ a birthday song.
Happy birthday
Birthday to you. Happy
Happy
birthday
birthday
to
to
you.
Happy
Happy birthday,
birthday,
EJ.
Happy birthday
Happy
to
Birthday
you.
to you.
Yay!
I was expecting some Malaysian or Philippines happy birthday song.
Oh yes, please, the Philippine, please sing. I have never heard that. Please sing.
It's
It's
the
the
same
same.
tune.
The same.
Really?
Just Filipino.
Yeah, please sing in Filipino, in Tagalog.
Oh, sige.
Yeah.
Maligayang bati
Bati sa iyong pagsilang. Maligayang, maligayang, maligayang
Maligayang
bati.
bati.
Nicer.
Good, Zera. Good thought. Yeah. So happy birthday.
Maligaya
EJ.
is happy birthday?
Maligaya is happy.
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 There's no birthday
Birthday.
translation in
There's one,
Oh.
pagsilang, in the next,
pagsilang.
Ah,
yeah,
the
pagsilang.
next,
The next line.
Okay.
So, EJ will send us burgers, virtual burgers.
An emoji.
Burger.
Yeah.
Yeah.
Okay. Thank you so much.
I am
Thank you so much.
49 today.
Yeah. Mhm.
Ah, wow.
Very young.
49.
Yes, 49.
Ay,
No,
Yes.
you're kidding.
Yes,
yes, I know.
Oh, you look so young.
thank you.
Oh, these people from Asia, they're so lucky.
It's maybe the hair.
Yeah, you're 49 because I'll be 48 next in this year.
In November.
We're only a year
1 year
apart.
apart.
Yeah, a year apart.
EJ.
Mhm.
Oh, go ahead. Sorry.
No, I just want to say I'm 52 this year.
Oh my God, that's not fair. I was born in the wrong continent. Honestly,
I'm 62.
no way.
Yes, way.
I think I should go home now. Bye.
How old are you? If you
I'm
mind?
38.
38.
And I really look 38, so that's not fair.
I envy you. You're so young.
No, I envy you. I envy your skin though.
Well, the webinar was great. The questions were great, much better than last time. It's so good. I think I'm gonna email some of them because I really don't know how the